Okay, we'll get started today. I get the distinct pleasure of introducing our speaker for our brain building seminar today. It's Dr. Michael Pizzi, who I've had been very fortunate to be able to work with him over the past couple of years on his projects and interest in TBI. And as I was mentioning earlier, one of the best things about introducing speakers is that you get to learn a lot about them that you didn't quite know before. For example, Dr. Pizzi had expertise in a minor in political science, and but it was early on in his education in Illinois, Northern Illinois University, that he also started his focus and interest in neuroscience. Moved on to get a master's degree at FSU. Then he got a PhD at the Medical College of Wisconsin. Then he went to Kentucky, and we didn't quite coincide because I did a stint there to do a postdoc with a very good friend and colleague, Dr. Kathy Satman. And there he started looking a lot about on TBI and getting exposed to spinal cord injury as well, and multiple forms of trauma that I suppose inspired him to pursue a degree in osteopathic medicine, again in Chicago. Right. Then, loving the Florida weather, he came back to UF and completed all of his training here. And it was really encouraging and exciting to see a lot of the work that he's done, moving on to a specialty internal medicine, neurology, leading the critical care unit in Jacksonville, working at the Mayo Clinic. And in talking to him in conversation separately, I've learned a lot about his interest in treating patients at a fundamental level. We don't have good effective treatments for individuals coming into the clinic with severe TBIs. And he's not quite exactly sure that we're doing the right thing. And he seems, in our conversations, extremely motivated to change course of treatment, standard of care. And I'm excited to see everything that he's doing. The list of training and awards that he has in his CV are extensive. He has substantial expertise, and you can see in all of his publications. And with that, I'll give the microphone to him.
All right. Well, thank you, Joe. And thank you all for coming and attending this. I really do appreciate, first and foremost, the funding from the Brain Center that actually got this pilot project off the ground, and then the work with Joe's lab, Jason Coleman and Marcelo Fibo, and we'll get started. So, like Joe mentioned, most of my time is spent across the street in the Heart and Vascular Neuromedicine Hospital. Our neuro ICU is on the 4th floor, and this is just stock photo. This isn't from our unit, but oftentimes we have patients with severe traumatic brain injury, even mild and moderate TBI end up in our unit sometimes. And a lot of the devices in this picture are giving us a hint about the physiology that's going on in this patient. They can't tell us what's going on. We kind of have to use these surrogates to kind of understand what's going on. And part of that is how we treat them medically. I'm a neurointensivist, so I only have medicines at my disposal, but our neurosurgeons, they can do certain surgical procedures. But fundamentally, we still don't know what's going on inside that person's brain after a traumatic brain injury and how our treatments may or may not be helping our patients. So these are just some representative CT scans of some of the acute neurologic injuries that we sustain or that we treat in our neuro ICU. But what's interesting is that with traumatic brain injury, you can get all of these different pathologies in one brain, right? You can have bleeding on the outside of the brain, an epidural hematoma or a subdural hematoma, the first two columns. You can have some subarachnoid hemorrhage where there's blood along the innermost lining of the brain. You can have contusions that hemorrhagically convert. There can be areas of ischemia that can also happen. So you've got multiple different types of pathologies within that cranium of a patient with traumatic brain injury. So I sort of approach the pathogenesis of traumatic brain injury as three problems, or the three Ps. One is pressure. All right, the brain is in a fixed skull in adults. In children, before the sutures fuse, there's still some flexibility, but within the adult, it's a fixed volume, about 1,700 milliliters in the adult brain, in the adult skull. And when we have an injury, whether it's stroke, TBI, there's brain swelling, but that occurs in this fixed volume. Some of the things that we can do, it's a fixed skull, we can call the neurosurgeons. They can take part of the bone off, a craniectomy, or we can address it with medicines like hyperosmolar therapy, mannitol, hypertonic saline. The other problem are the pipes, the blood vessels. And this is really where I'm going to zone in with my interest in the blood-brain barrier and how changes in that can affect the rest of the brain physiology. In particular, how it regulates blood flow into the brain, because ultimately it comes down to the third problem, power. We got to get blood, which contains glucose, oxygen, lactate, ketone bodies, alternative energy sources. So we got to get a way to have the fundamental building blocks of the brain to that tissue that needs it after injury. And that's all deranged after traumatic brain injury. And why is the brain vasculature so important? Well, in the adult, there's about 400 miles of length of vasculature in the adult brain. And if you take all those vessels, it's about 2.5% of the total brain volume. And there's some regional variability depending on the area that that brain serves. So white matter is not terribly metabolically active, so it doesn't have that high density of blood vessels, whereas highly metabolic areas like the hippocampus have a higher density of blood vessels. So I would argue that pipes are really the things that affect the pressure and the power, the other two P's in our three problems. And just to give you another in vivo illustration of how important these pipes are, this is a 2-photon microscopy Z-stack image of blood vessels from Andy Shee's lab in Seattle, just demonstrating just how complex and how densely packed all these blood vessels are. And it's tremendous when you think about each neuron is only about 15 microns from the nearest capillary. So tremendously important how getting blood to these cells helps with recovery. Part of the vasculature that I'm interested in is the blood-brain barrier. Lots of different components make up the blood-brain barrier. The innermost part of the blood vessel is the endothelial glycocalyx. I'll talk a little bit about that in another slide. Next to that, so extending off of the endothelial cells is the glycocalyx. Then you have your endothelial cells. So it's just a one-layer endothelium that lines the lumen of the blood vessel. That's encased in a basement membrane consisting of collagen and different proteoglycans. And then pericytes, which are these very unique dynamic cells that wrap around arterioles and first and second order capillaries, and they do a lot of the regulating of blood flow into the capillary bed. And then lastly, astrocyte end feet. It's about 95% of the capillaries have astrocyte end feet attached to them. They regulate a lot of water going in and out of the brain, very important with cerebral edema after traumatic brain injury. And Martin Lauritsen published a paper in 2017 demonstrating more of a tripartite blood-brain barrier based on molecule movement across each of these different layers. So the first layer, the glycocalyx, allows molecules up to a certain size through, and then the endothelial layer, and then lastly, the extravascular compartment, all based on molecular size that these compartments allow through. And then just to give you an example of the 2-photon, which we'll get into in a little bit, the red here is the endothelial glycocalyx. The green is labeled dextran, demonstrating the lumen of the blood vessel. And you can see this beautifully layered endothelial glycocalyx around the lumen of the blood vessel. And this, I would argue, is the first line of defense of the blood-brain barrier. And I'm very interested in how it changes after injury and things that we could possibly do to promote its recovery and function. So the glycocalyx, like I mentioned before, are these sugar molecules that project from the luminal aspect of the endothelial cells into the lumen of the blood vessel. And glycocalyx stands for sweet husk. And why is it sweet? Well, most of these molecules of the glycocalyx are proteoglycans. They are made up of core proteins that are embedded in the endothelial cells, and then these repeating disaccharide units that go on and on and on. They have variable lengths, they have variable levels of sulfation, so they're very negatively charged molecules, and they intertwine with each other. And the most common proteoglycans are the glycocalyx, syndecan, which is a heterogeneous group of chondroitin sulfate and heparan sulfate molecules, glypican, which is a heparan sulfate proteoglycan, and then hyaluronic acid, which is very unique. It's just these repeating disaccharide units, and they are anchored to the cell membrane by their interaction with CD44. So these sugar groups create the first line of the blood-brain barrier. They can bind a lot of water molecules. They can bind growth factors. They play a role in how blood flows through, or the rheology through different capillary beds. And they can also detect the shear stresses of blood going through there and relate that mechanotransduction to the cytoskeleton of endothelial cells, again, helping regulate a certain amount of blood flow into the capillary bed, because the brain only wants to see so much blood at one time, not too much and not too little. There's this fine tuning of the cerebral blood flow that's mediated in part by the mechanotransduction through the glycocalyx. After injury, whether it's stroke, like this figure shows, or TBI, there is this biphasic change in the permeability of the blood-brain barrier. And we also see that permeability correlates with the shedding of two of the main constituents of the glycocalyx, hyaluronin and syndecan. And so this blue line represents the expression of these molecules of hyaluronin and syndecan on the endothelial cells, and how as that expression decreases, the blood-brain barrier permeability peaks. And then there's a little bit of recovery, and then there's another peak about a week after injury or the ischemic event. And again, it very tightly correlates with the expression of these molecules in the glycocalyx. That's important because how we treat brain swelling after stroke or traumatic brain injury, medically, we use hyperosmolar medications like hypertonic saline and mannitol. So this is just a cartoon demonstrating this osmotic gradient by giving a hypertonic saline solution, all these sodium molecules, that creates an osmotic gradient between the lumen of the blood vessel and the brain tissue extracellular space, creating an osmotic drive for the water to go from the brain into the vasculature and then hopefully decrease the cerebral edema. But the big but is it's predicated on an intact blood-brain barrier. If you don't maintain this blood-brain barrier, these sodium molecules, mannitol, can get into the brain and could potentially cause a reverse osmotic gradient and actually exacerbate the brain swelling you're trying to treat. And that's why in the clinic, we can see that after a certain period of time, patients don't respond to these medications. And that's probably why the blood-brain barrier has degraded. There's no more osmotic gradient, and we don't have a whole lot of options at that point in time to treat their increasing pressures in their heads because of the edema. So my area of research really centers on the blood-brain barrier, in particular the proteoglycans that make up the blood-brain barrier. And we see this with edema. And eventually, if you carry the breakdown of the blood-brain all the way to its final termination, you get this hemorrhagic transformation where there's this complete disruption of the blood-brain barrier and blood can get into the brain. And so work with Joe's lab looking at a repetitive mild TBI model, and Dr. Candelario Halil's lab with stroke. We've been looking at some of these changes with 2-photon, and I'll show you that data. There is some evidence that proteoglycans, since they are detecting that shear stress, can affect cerebral blood flow and cerebral autoregulation. We tried doing some of this in Joe's lab with the 2-photon, and I'll talk about that. That's to be continued. And then lastly, I don't have time to discuss the role of proteoglycans in brain compliance. You can think of compliance as sort of the squishiness of the brain. It's almost a Goldilocks situation where you don't want too much compliance or too little compliance because that affects cerebral blood flow. And been trying to develop a brain phantom model with Dr. Christine Schmidt, Lee Murphy, and Dr. Wong's lab that's ongoing as well, but we won't touch on that in this talk. And then these are all interrelated. So these are not things that happen in isolation. They're all interdependent on each other, how the blood-brain barrier affects blood flow, cerebral edema, and then the compliance of the brain tissue itself, which feeds back on to the blood vessels. So very intricately related. So I'm gonna spend a good chunk of my time talking about two-photon microscopy. So this allows us to look at in vivo in a mouse model, the cerebral vasculature in real time in a living mouse. What we do is, or I do, on days I don't drink a lot of coffee, I create a thin skulled window about 15 microns in thickness over the cortex. And then we have a mount. We've modified this mount. This was stolen. This cartoon was stolen from Andy Shee's paper. But we initially used this head mount, modified it to a little more stable one. And then we were able then to put a glass cover slip over that thin skulled region, and then under anesthesia, get our measurements with the two-photon microscope. And I don't know if you have a lot of experience with two-photon, but I'll just briefly touch on why this is such a powerful tool. So two-photon, you get two photons basically coming simultaneously to hit a fluorescent molecule. The probability of two photons simultaneously hitting one fluorescent molecule is incredibly very high. So what you have to do is you have to give multiple pulses of light in order to increase the probability of two photons simultaneously hitting that fluorescent molecule. And we do that by having pulses so rapid they are on the order of femtosecond pulses. So they're only about 100 femto. We do 100 femtosecond pulses, and there are about 80 million of them per second. So the duration is very short, very rapid pulses, which means that the power, if you were to just shine light onto this tissue or organ that you're looking at, in order to achieve the probability of two photons, you would have to have over 100,000 watts of power onto that tissue. You would obliterate the tissue. But by doing multiple pulses, you use less energy, usually only about a watt of energy. That allows the no bleaching of your fluorescent molecule, and it also doesn't cause as much tissue injury because of that lower energy. So it allows us using longer wavelengths to get deeper into the tissue, about half a micron in depth from the surface. And then your area of absorption is extremely small. So we have a very focal area. You don't have all the refractory light coming off because there's just two photons simultaneously hitting that fluorescent molecule. So you get exquisite resolution. You don't have as much tissue damage, and allows you to do all this in a living organism. This is our two-photon scanning microscope that we have just around the corner here. This was before Dr. Coleman put this really nice dark screen all around it to knock out any ambient light. So this is our setup just around the hall. And what we do is, with the help of Dr. Ravi in Joe's lab, is we utilize the Chimera repetitive mild traumatic injury model. So it allows us to use a closed head injury model where the mouse is laying supine here, the head is over this silver plate, and then a pneumatic piston comes up, causes an injury to the brain, or it causes an injury, but not sufficient enough to cause a skull fracture. And we do this under anesthesia. We do 2 hits, so 24 hours apart. After the second hit, I take that animal, I do the thin skulled window here, put the glass cover slip on, and then do a retroorbital injection of a fluorescently labeled dextran, and then image them with the 2-photon microscope. And again, this is the setup as well. So the first thing that we did is we wanted to look at, is there any changes just in the capillary diameter as a function of depth? So we can start at the cortical layer and then go about 300 microns in depth. And we color coded the depth here to kind of give you this Z-stack orientation so that we can look at the different layers of the cortex and see if there's a change in the diameter of these capillaries as we change the depth. And we do that by Fiji. We do these bisecting marks throughout multiple layers, and then can look at the diameter of the different capillary beds. And what we found is that in red are the TBI, and then the black are the sham. So they undergo anesthesia, but no traumatic brain injury. And there is interestingly, larger diameter of capillaries near the surface of the brain. And as we get deeper into the brain tissue, we don't get as much of a difference in the diameter of these capillaries. So that was our first foray into using 2-photon. Then really what we're most interested in is there any permeability of the blood-brain barrier? And we measure that by perivessel fluorescence. So this is a mouse that received a fluorescently labeled dextran, which will illuminate the lumen of the blood vessel. And then with Dr. Jason Coleman, he created this plugin to Fiji where we're able to create this rectangle where we're measuring the perivascular fluorescence, and we have a standard rectangular size for each vessel. And this is based on the concept of a full width half maximal. So in order to identify what is blood vessel and what is perivessel space, when you get the maximum fluorescence, if you take that half max, that defines the border of your blood vessel of interest. And then everything outside of this full width half max is your perivessel space. So this blue area here is this perivessel region that we're going to measure the intensity of fluorescence. And when we do that, we can see that fluorescence actually increases as we get deeper into the brain tissue. So the inverse of the vessel diameter, where vessel diameter was large and superficial, now we have greater perivessel fluorescence as a marker of blood-brain barrier permeability the deeper that we get. And we're kind of maxed out at 300 microns from the cortical surface, just from the limitations of the microscope. And you can see how that compares to the sham mice, where there's still some perivessel fluorescence. That's not unexpected, but at the deeper layers, you don't see nearly as much perivessel fluorescence. So there's definitely a perivascular permeability to the blood-brain barrier that we can detect with 2-photon in our TBI mice. So this compares to probably the closest study out there where they use, this is Schwarzmeyer's lab in Germany. They use a controlled cortical contusion TBI model. So this is different. This is, you make a hole in the skull and a piston goes into the brain parenchyma and causes a focal injury into the parenchyma. And then they use 2-photon microscopy and then looked at perivessel fluorescence. And Not surprisingly, they had a little bit more fluorescence, more superficial than deep, but clearly the closer to the injury, which is A1, and further from the injury, A3, there's this gradient of blood-brain barrier permeability the further you get from the site of injury, but it also is a little bit more prominent, superficial than deep. And then this was an interesting paper from a stroke model where they looked at blood-brain barrier permeability along the arteriovenous axis. So is there a difference in blood-brain barrier permeability at the arterioles versus the capillaries versus the venules? And interestingly, what they found was that venules had a greater amount of perivascular fluorescence as a marker of permeability, which is rather interesting. We don't usually think too much about the venule side of the AV axis, but this may be of interest in TBI. So I went through our data and looked at these changes along the arteriovenous axis. And what I found was that the greatest amount of perivessel fluorescence was at the level of the arterioles, and then still a little bit more compared to shams in the capillaries. And by the time I got to the venules, they were very similar in their perivascular fluorescence compared to shams. So a little bit different than in that stroke model where they saw more perivascular fluorescence along the venules, whereas I see it more along the arterioles and capillaries. And then how does the blood flow at different points in the arteriovenous axis affect or possibly affect blood-brain barrier permeability? So this was an in vitro model that was developed recreating an arteriole into a capillary, and then they could measure the flow dynamics, and then they lined the vessel wall with endothelial cells to sort of recapitulate a capillary. And then they were able to measure these forces through there, and then also the fluorescence where it leaked out. So as the blood is coming down this arterial, quote unquote, there's this near wall and then this far wall, and then down here where the flow is static, and they measured the perivessel fluorescence and found that, you know, right at that bifurcation, there is a little bit more fluorescence that leaks out from this endothelial monolayer. And so I thought, well, is there something similar like that in vivo? And if we go back and measure the perivessel fluorescence at the arterial and the capillary immediately post bifurcation and then mid capillary, is there a difference? And there was a difference when we look at the immediately post bifurcation. So again, it's an arteriole that bifurcates into two capillaries, measure the perivessel fluorescence at that point, and there's significantly more fluorescence compared to shams. And then interestingly, mid-capillary, not quite as much perivessel fluorescence compared to shams. So again, not surprising given the flow dynamics immediately coming off an arteriole into a capillary bed, there seems to be a little bit more blood-brain barrier permeability at that point. How is that affected by the endothelial glycocalyx? So this was work when I was in Andy Shee's lab. We were testing different types of lectins to see which would bind the endothelial glycocalyx the best. And what we found is this Lycopyrrhizine esculatum agglutinin, the LEA, tends to bind more robustly. Again, the endothelial glycocalyx is a very heterogeneous conglomeration of glycoproteins, and LEA tends to bind best. And of course, it's been validated in an independent lab. But this was just some 2-photon images from Andy's lab. And you can see how nicely the endothelial glycocalyx stains with the LEA lectan. And then there's a Texas red labeled dextran to illuminate the vessel. So future work will look at the thickness of this endothelial glycocalyx and how does that correlate with areas of perivessel fluorescence. So we'll see, hopefully that'll happen in the next year or so. The other thing that we were interested in with 2-photon is being able to measure blood flow through these capillaries. So what we tried to do is measure red blood cell flux. So if you can make out these dark bands through these capillaries, every time you see that dark band, that's a shadow of a red blood cell passing through that capillary bed. If you measure, you can measure the velocity through a segment of that capillary based on that. Oh, okay. And what you can see is that there's the segments that you can measure, these line scans, where you can measure the velocity of this red blood cell. And that's called the flux. If you know the diameter of the blood vessel at the time, then you can measure the blood flow. But we were just trying to measure the flux of red blood cells through these capillaries in mice subjected to repetitive mild TBI versus sham. Unfortunately, we're having a difficult time with the data coming off the scope and the computer crashing, but theoretically, it can be done. And this is just another description of it. Here's your line scan measuring the movement of red blood cells through this section of capillary. And if you take the diameter of the capillary at the time, you can measure the cerebral blood flow through that vessel. But we just have to make some adjustments on our computing power. But that's another project waiting to be done. Now, there is a resolution limitation to 2-photon microscopy. As you can see, the 2-photon, optimistically, theoretically, gets to about 1,000 microns in depth. We were more around 300, which gets you to about layer 2 and 3 in the mouse cortex. So for all intents and purposes, we're at this very superficial layer of the mouse cortex. This is a closed head injury model of TBI, and it's not a focal injury. We wanted to know, are there other deeper brain structures where the blood-brain barrier may be affected? And so that's why we turned to a MRI sequencing scan called dynamic contrast enhanced MRI. And could not have done this without a lot of help from Dr. Fibo, Ravi, and Zhang from the Magnet Lab. And what it really is, it's a MRI sequence where animals are injected with gadolinium contrast. And you're measuring the rate at which gadolinium leaves the capillary and goes into the extracellular space in the brain tissue. This rate at which it leaves the capillary and into the extracellular space is called the K-trans. And so that would allow you to measure blood-brain barrier permeability through the entire depth of the brain, not just restricted to that superficial layer. So again, with the help of Dr. Ravi doing the repetitive mild TBI, we did that twice. After the second injury, we put the mouse in the 7 Tesla scanner just down the hall. And after a lot of technical tweaking from Dr. Fibo and his lab, we were able to measure the rate at which gadolinium goes into the extracellular space. And so we could see that it peaks about 10 minutes after the injection of gadolinium and then tapers off pretty rapidly after about half an hour. And then these are the pre-gadolinium scans. And then after gadolinium, calculating the K-trans, we were able to see these areas of extravasation of the gadolinium throughout the entire depth of the brain, which is not surprising. This is a closed head injury. There's a lot of rotational forces that act throughout the entire thickness of the brain. And so just to kind of reorient yourself, we were just looking at this very small area of cortex, about 300 microns in depth. Now with dynamic contrast MRI, we can look at BBB permeability throughout the entire depth of the brain. And we are seeing quite a number of areas affected with this repetitive mild TBI injury. This is incredibly important because this is a technique that you can do in humans. You can't do 2-photon microscopy in humans, but we do DCE MRI in humans. And so this is something that could be directly translatable into the clinical side of things, to look at this as a modality to measure BBB permeability. And then lastly, is there a way that we could possibly repair the endothelial glycocalyx after injury? So this was a project that actually I did with just some test mice with Dr. Candelario Halil, where we did a ischemic stroke model in a mouse, occluded the middle cerebral artery, and then after occlusion, we fed a catheter into the internal carotid artery and infused a high molecular weight hyaluronic acid that was labeled to Pizzi. So it was a way that we could introduce exogenous hyaluronin that could incorporate itself into the glycocalyx and hopefully minimize some of the damage to the blood-brain barrier that occurs after stroke. So what we did, this is a image after Eduardo put a filament up the internal carotid artery, occluded the MCA. He took out that filament after 45 minutes of occlusion, and then we ran this catheter into the internal carotid artery until we met some resistance, and then we slowly infused high molecular weight hyaluronin. The reason we use high molecular weight hyaluronin is that it's actually anti-inflammatory. Smaller versions of a hyaluronin are known to be quite inflammatory, and so that's why we chose that. And then we're able to demonstrate using 2-photon after that infusion that the high molecular weight hyaluronin actually bound to the lumen of the blood vessels. And then when we co-label that with Texas red dextran to show the intravascular space, we can see that our exogenous hyaluronin bound to the blood vessels. And could this be a potential way of shoring up the blood-brain barrier? That story is still to be told. So at least we were the first to demonstrate that after stroke, you could infuse exogenous hyaluronin, and it does bind to the vasculature. All right, so just in conclusion of these studies, we found that there's an increased diameter of superficial cerebral blood vessels following repetitive mild TBI. Perivessel fluorescence increases with the depth from the cortical surface following TBI. The greatest perivessel fluorescence and BBB permeability was associated with arterioles and capillaries, not so much with the venules. And there's a trend towards increased perivessel fluorescence in post-bifurcation capillaries. So where do we go from here? A lot of our patients do develop cognitive dysfunction after an acute brain injury, whether that's stroke, TBI, and part of it is this right here. What is the cerebral vascular dysfunction? Is it the blood-brain barrier? Does it allow certain molecules and cells to get into the brain that creates collateral damage? Is there this long-term effect on cerebral vascular dynamics, cerebral blood flow, cerebral autoregulation? Is there damage to the endothelial cells that are no longer able to utilize energy? Is there injury to the mitochondria? Does that affect the glycocalyx synthesis? How does that affect it? So there's a lot of open questions. How does a traumatic brain injury, even a mild traumatic brain injury, ultimately increase your risk for neurocognitive disorders down the road. So like I mentioned earlier, what I want to do next is measure the endothelial glycocalyx and how does that change in vivo correlate with blood-brain barrier permeability? How does that affect cerebral blood flow dynamics? And then ultimately, how does that also affect the cognitive recovery after traumatic brain injury? So hopefully we'll get some studies looking at binding of LEL lectin to the glycocalyx, measure that, and see how does that associate with perivessel fluorescence. We can also, this study, also I don't have that figure here, you can actually degrade certain aspects of the glycocalyx. There are enzymes you can give in vivo that degrade the hyaluronan, the heparan sulfate, the chondroitin sulfate. So you can basically shed certain components of the glycocalyx, and how does that affect blood-brain barrier permeability and cerebral flow dynamics? And then how does that affect cognitive function in the long term? So with that, I am, and here's basically a overview of where we can go with mice, but also you can measure this in humans. So in mice, we can look at BBB permeability, blood flow, neurovascular coupling, all with 2-photon microscopy. We can measure the molecules of the glycocalyx as biomarkers of endothelial glycocalyx damage. How does that relate to their cognitive function? And then, as I mentioned, with humans, you can also measure blood-brain barrier permeability with DCE MRI. Neurovascular coupling you can do with transcranial Dopplers. So the things that you can do with a mouse, with 2-photon, there are analogous modalities in human patients. And how does that affect their cognitive function down the road? And with that, I thank you for your attention.
Mike, I have a question.
Yes.
So it seems as though the permeability increases with depth after the injury. Do you see in your data that it increases linearly, there's an exponential, or does it come back at some point within those 300 microns?
So it continues linearly. And so the deepest we could get is about 300 microns, and it kept increasing as we were going deeper.
Given your expertise in the clinic, do you think that this will continue on to deeper structures, ultimately affecting massive, big blood vessels, like even the circle of Willis?
I don't know, because there are different mechanisms that could affect those larger vessels. So whenever that second slide with some of the blood along the surface of the brain, the subarachnoid blood, the subdural blood, that can affect those larger vessels. So it's hard to say what's actually affecting blood flow dynamics in those vessels. Yes, Luis. That's a great question. I use 45 kilodaltons. Again, going back to Martin Lauritsen's paper from 2017, where his lab showed what molecules permeate, and these are in uninjured mice, each layer of the tripartite BBB. Certainly 45 kilodaltons would get through the glycocalyx and the endothelial cells. So after an injury, of course, now you also have to get through the basement membrane. You've got all these astrocyte end feet there. And most of other labs use 45 kilodaltons as sort of the standard molecular size to measure BBB permeability after an injury, whether it's TBI or stroke.
The other question that I have is, what is the relationship between photo so there's pathway that is activated. Also, in addition to that, you know that of course you have a trauma is okay. So you have some system and usually buying connecting out. So, of course, but I'm not my question is very broad, but I want to know what is known in this setting. So what is the association of the medical practice specific this guidelines both more
With the tight junction molecules? Mm-hmm.
also, I want to know if anybody knows about this.
So we know that though the proteoglycans are connected through integral proteins in the cell membrane to the cytoskeleton. Those cytoskeleton proteins do connect to the tight junctions, and there is some evidence that indirectly they cause the breakdown of those tight junction proteins. So it's almost like you need some tension in the system. And if you damage those proteoglycans of the glycocalyx, they don't transmit that mechanotransduction intracellularly to those tight junctions. And then they don't have as great a strength in connecting adjacent endothelial cells. And we do know that the same molecules, the matrix metalloproteinases that degrade the proteoglycans, also degrade those tight junction proteins. So it's hard to say, is it the damage to the proteoglycans that causes the tight junctions to start to pull apart? Or is it also in conjunction that there's also the same enzymes as breaking everything down, breaks down those tight junction proteins as well?
They ask you because we're very interested in the graphic show of the situation of the expression of those life. And I ask you because, at least in our model, we have some the expression of tight junction at the same time. And you can see that at the beginning of the infection, they disappear, and then they come back. So perhaps they're not the same time from the top, but there is that that pattern that we very hard to understand.
Yeah, and it's of course more complicated than that, because a paper just came out, I think last week, where lactate, which we know after TBI, most of the ATP is generated from lactate, not glucose. That lactate actually it correlated with the internalization of tight junction proteins. So. Mhm. Mm-hmm. Mm-hmm. Yeah. Anisha? Oh.
Great talk, Mike. Good to see your work. One question. So I know in the world of cartilage fighting index, HA is the thing, right? Have you gleaned any inspiration on how HA is typically the targeted for proteoglycan, that there was something HA in that, or some of those others, that there was something to enhance to prevent cardiac degradation in the knee? So I'm not sure if there's any inspiration from that space, but I just wanted to chime in that I think that might be something to consider looking in that space, the osteoarthritis type space. Also, to Joe's point, as the depth decreases, there's increased is that based on, because you have one type of modality? I don't know if you've seen this in other spaces, but how would that look in an overpressure of lash trauma injury versus a blunt trauma injury? I think, was it directly away a certain distance from the injury site, or was it just randomly in some random death?
So the injury site, so it's a closed head. So the piston will hit the intact skull as midline as we can get. We don't reflect back the scalp, so we just put the head about as midline as we can over the piston so that it hits right along bregma and that it hits as midline as possible. So what we're measuring is really going to be lateral to that point at which the piston hits, because we can't make a window right over the suture because of the venous system. So we're a little bit lateral to it. In other injury models of closed head injury, so like a weight drop apparatus, they have shown BBB permeability throughout the entire depth. I have not seen that with 2 photon, a closed head injury model. So this is relatively unique. Most of it's with the controlled cortical contusion, and then fluid percussion, which is also, you need a craniectomy for a focal injury as well. It's supposed to mimic a diffuse injury, but it's a focal cranie.
All right, can I just add to this conversation?
Yeah.
Discussion? One thing that's unique about this model is that the head isn't fixed, so you're getting a lot of rotational.
Yeah.
So the question is, how much is this rotation then affecting blood flow and the integrity of the blood vessel?
Mm-hmm.
And that is a unique opportunity that is available here because of the difference in the model.
Yeah, that's why I think the MRI was so informative when we saw how deep the permeability went. I mean, it goes through the entire depth of the brain, that we weren't really paying attention to or couldn't measure with 2 photon. So that, yeah. But also to address your first point, there is a lot of interest in proteoglycans in the glomeruli of nephrons. And there's actually an FDA approved, not FDA, European approved, it's not approved here in the United States. It's a commercially available gamish of heparin sulfate and chondroitin sulfate proteoglycans that has shown to improve the endothelial glycocalyx of the glomeruli of nephrons. So there is some commercial interest in giving exogenous proteoglycans to bolster the vasculature, the endothelial glycocalyx. So, yes, sir. Mm-hmm. I love that question, and I think that was part of the discussion. The FDA approved the first monoclonal antibody against the recommendation of the Scientific Review Committee. So I'm not going to weigh in on those waters, and there are experts here. Whether or not this is an amyloid or tau, I don't want to venture into that. But I think this is a really good point that you raised, that the flip side is, especially in neuro-oncology, how do we get things into the brain across an intact blood-brain barrier? And if you go back to the original papers describing the blood-brain barrier and how they measured it, using dyes that were given intravascularly, they opened the BBB by giving mannitol and hypertonic saline, the same things I give to my patients that actually causes the opening of the BBB. And so, there's there's a lot we don't really understand in how things get across. And yeah, monoclonal antibodies, maybe IgGs, IgM, certainly not. They're pentamers. There's no way they're going to cross the blood-brain barrier. So yeah, I scratch my head about that. That's a good point. So again, in Andy Shee's lab, he has a pediatric neurologist that works in his lab, and she has shown that CAR T cells, which are basically chimeric cells, they are not meant to cross the blood-brain barrier. But what they're finding is for other cancers, they are using them in small groups off-label for glioblastoma cancer cells. How they get across the BBB, I'm not entirely sure, unless the cancer cells themselves are dumping out all these matrix metalloproteinases. They're creating the environment where the BBB could be permeabilized. But what they're actually finding is that there are lots of capillaries that are occluded after giving a CAR T therapy. And we see that clinically. We do see a lot of ischemic strokes in patients on CAR T. We don't know the mechanism by which that's causing it. But some of Andy's work shows that there is damage to the endothelium, and in particular, pericytes somehow are affected, and that could be affecting blood flow through these capillaries. I think it's multifactorial, but it's to answer the question simply, yes. If they have looked at the glycocalyx in post-mortem samples, you can look at it with electron microscopy. You can see that the glycocalyx is reduced, that there is lots of small vessel disease associated with Alzheimer's and Alzheimer's-related dementias. Part of that is the glycocalyx is disrupted in those affected vessels. So I think there's a fair amount of evidence that they are related. And I think I just want to just double check the chat. Okay, never mind. All right. Well, I appreciate everyone sleeping.
I have a couple questions. Is there any, can you comment on any potential interventions that might help remodel the intercellular space? And I'm thinking about other risk factors, comorbidities, things like that. Some of the data that we have recently, and you've been helping us with that, is how age is one of the major driving factors that affect recovery from injury, even more so than TBI itself.
Yeah. What I find really fascinating, and again, to kind of tie into Lakeisha's point, is hyaluronic acid. So the extracellular space in the brain is about 25% of the total volume. Hyaluronic acid binds a tremendous amount of water molecules. That creates space between the matrix proteins, right? If you dehydrate the hyaluronic acid, it collapses on itself and you eliminate a lot of that space. And we now know how important the lymphatic flow is to kind of wash out a lot of these extracellular proteins that may be contributed to cognitive dysfunction. So yeah, I think that extracellular space has been ignored. And is there a way to increase the binding of water in non-pathological states to keep that space open to allow optimal lymphatic flow? And I think Niedergaard's lab at University of Rochester has been showing a lot of this lymphatic flow with 2-photon, and that as that extracellular space diminishes, the lymphatic flow diminishes.
Thank you very much. I
Yeah.
appreciate it.
Thank you all.
I would say